Hello students, in previous lecture we discussed the outline of our syllabus of paper, that is a bioinorganic and medicinal inorganic chemistry. Now in this lecture we are going to discuss some important and basic concepts in bioinorganic chemistry. Bioinorganic chemistry is a very fast developing branch of chemistry. In that, metal enzyme play a very important and vital role. Depending upon the quantities of metal in biological systems, there are two types of metal, that is a bulk metals and trace metals. Along with this lecture, we are also going to discuss what are the cofactor, coenzymes, what are the metalloenzyme, their functions, and prosthetic group and substrate molecule. This all concept we discuss in detail one by one. First, we start with the bioinorganic chemistry. Bioinorganic chemistry is the study of both natural phenomena, natural phenomena such as a behavior of metalloenzyme as well as artificially introduce metal ions which are non-essential in medicine and toxicology. So what is meant by the bulk metal? The metal ions which are present in biological system in bulk quantities is known as bulk metal. The example of bulk metal, that is sodium, potassium, calcium. These are the bulk metals. Bulk quantities means in large quantities. The metal ions which are present in large quantities in biological system is also known as a bulk metals. Now what is meant by the trace metal? Trace metal, the metal ions which are present in biological system in trace amount or small amount is known as trace metal. Example of trace metal, that is Fe, copper, cobalt. These are the trace metals. Now what is meant by the metal enzyme? Metal enzymes are nothing but the metal containing enzymes enzyme containing metal ion which are directly bound to protein or non-protein component is known as metal enzyme. Now, what are the functions of metal enzyme? The first function of metal enzyme is that it will bind to substrate by binding to substrate to orient properly for a reaction. Then next, by mediating redox reaction through reversible change in metal ion oxidation state. And the third function, that is by electrostatically stabilizing negative charges. So these are the three important functions of metal enzyme. Now what is meant by the coenzyme? Or cofactor. Cofactor is a non-protein chemical compound or metallic ion required for enzymes activity. Is nothing but the cofactor, or we also define that cofactor is a helper molecule which assists in biochemical reactions. So example of cofactor that we can take as an example of ATP. ATP is a unique ability to drive chemical processes in biological systems such as to transport protein and to increase the enzyme's activity. Now cofactor are two types, that is organic cofactor and second, that is inorganic cofactor. The example of organic cofactor, that is a heme, some flavine, heme, and inorganic cofactor have a magnesium ions. Now, what is meant by the apoenzyme? Apoenzyme is a protein that form an active enzyme by combination with coenzyme and determine the specificity of system for a substrate. Now, what are the example of apoenzyme? The example of apoenzyme is a holoenzyme which polymerize DNA and RNA Holoenzyme polymerase DNA and RNA which have multiple protein subunits. So now next, what is meant by the prosthetic group? Prosthetic group is a non-protein compound, non-protein group or compound forming a part or combined with protein component. is known as prosthetic group. The example of prosthetic group is biotin, again flavin. Now what is meant by the substrate molecule? The molecule on which enzyme acts is known as substrate molecule. So these are the important and basic concepts in bioinorganic chemistry. Now in next lecture we are going to discuss the metalloenzyme of zinc, that is a carbonic anhydrase. Thank you.